My project focused on investigating the secondary role of benzoxazinoids. Benzoxazinoids are primary defense compounds found in grass species that are currently understood to function in herbivory and pathogen defense, or they play a secondary, lesser understood role in regulating other metabolite networks in the cell. So my mentor identified two compounds that are positively regulated by benzoxazinoids, Catechol-Glucoside and acetylcatechol. And she identified 4 possible genes that could be involved in its synthesis. So my job was to localize these genes as a way of understanding the function of the compounds. But why not just localize the compound? It seems much more direct. It's not really possible to localize the compound directly. We can isolate it, which I do later in my poster, and I'll explain, but it's not possible to trace it in the cell. So my method for localizing the genes was tagging them with something called a GFP. That stands for green fluorescence protein. And essentially it allows the gene to fluorescence in the cell. And judging by where it is in relation to the cell organelles, we can identify its function. So we inserted the gene into Agrobacterium, which is a bacteria that's able to infiltrate our model plant, N-benthamiana. And we used N-benthamiana instead of Maize because Maize is very difficult to infiltrate with any sort of bacteria, so this made it much easier for us. And then we performed protoplast isolations, which is essentially where we dissolve the cell wall of the cell, and you end up with this circular cell as opposed to kind of the scribbly cell that N-benthamiana is usually in the leaf. And this allows us to see better fluorescencing, a better picture. So with the results, we saw 2 different kinds of fluorescencing. We saw fluorescencing in the protoplast, in the chloroplast, and in the cell wall. So we don't necessarily know what that means. It could mean that it's localized in 2 different areas, or maybe there was an error in our experiment. So we'll have to really investigate that. Another thing ideally, the original goal of this project was to localize all of the genes at the same time. We aren't necessarily certain which genes are actually involved in synthesis, since these are candidates. So one step I performed in co-localization, towards the goal of co-localization, is a PCR mutation on the acyltransferase gene, which is the one that converts Catechol-Glucoside to acetylcatechol. And essentially what we did, there was a stop codon which was preventing fluorescencing. It's just 3 base pairs right after the gene, in between the GFP. And we flanked this with primers. And essentially we created 2 replicate vectors that excluded the stop codon. So this was not successful for us, but this is the method we would use in the future to co-localize the genes. So as far as outlook goes, we are looking to better the fluorescence picture through techniques like Z-stacking and dyeing the cell organelles. So Z-stacking is a technique where you use a laser to individually identify the layers of a cell, and that allows us to get a 3D image of the cell and see where within that 3D space it's localizing. And dyeing, we just, again, kind of attach not necessarily a GFP, but maybe a YFP or a different color protein to a cell organelle, and that allows us to see whether it's localizing within that area. This is a side project that I did with the Schroeder lab, which is a chemistry lab. We don't necessarily know the structure of our main compound, since this is an entirely new compound in Maize. So part of my job was to identify this structure through something called NMR. So NMR stands for a proton spectrometer. And essentially this allows us to identify the individual protons in the structure and map those to the surrounding chemical environment of the cell. So depending on the electronegativity of the surrounding atoms, it kind of spreads out the protons over the x-axis. So this is the first step. We'll perform other types of proton spectrometry to map the protons to certain carbons and really map out the structure of the cell. So main takeaways, this project isn't directly applicable to farms at the moment. However, this is increasing the database of knowledge surrounding benzoxazinoids, which we know are involved in plant defense against pathogens and herbivory, so they're obviously very important to the survival of the cell. And hopefully at some point this information will be able to help farmers perform mutations to create more resistant plants. I guess I had this image of research in my head that was totally out of sync with the day-to-day work. I guess it's much more repetitive than I thought. You really follow certain procedures over and over, and I always thought that working in Cornell, you might some of the procedures might be more experimental and everything would be new, and there really wouldn't be any patterns. But I've kind of taken comfort in realizing that there's things you can master and take with you throughout the program.